Are we this way? Did it start yet? Oh, hi out there. So this is honors bio, and we have 3 goals today. We're going to review the cell cycle, mitosis, and meiosis. Those are the 3 things. So hopefully you are in PEIJVD. Okay, and the first thing I want to give you is like a little list of things to know. So take a look at that and see if you feel comfortable with the things to know. Move me over here a little bit. And I would probably at this point just highlight the scary parts, if there are any scary parts, which I hope there are not. But highlight the scary part so you can just focus on that as we go through the review. Okay, do you need any more time? Can I move forward? All right, because you will have this, right? I will send this to you, or you have it right now. All right, so next, computer struggles so hard when I try to do multiple things. Hang on, sorry. Come on, little computer. Sorry, delay of game. Right here, cell cycle. Okay, so the cell cycle, what you're going to want to remember is that you have I for interphase, right? And that's all of this is interphase. And then you have M. And within the M, you have mitosis and you have what? Cytokinesis. But it could just as easily have been meiosis and cytokinesis. All right. The M is just, remember, both mitosis and meiosis are only focusing on the nuclear division, what's going on with the little chromosomal shuffle as they move around and you break down the nuclear envelope and all the process of separating the chromosomes and rebuilding the nuclear envelope. That is mitosis and meiosis. Cytokinesis is everything else in the cell. Now, interphase, remember, has 3 parts to it, G1, S, and G2. Okay, and remember what happens during G1, and always remember G1 follows mitosis, right? So after we're going to just go with mitosis, all the cells would be very what? Small. So what are they going to do in G1? They're going to grow, and you're going to make more organelles. Okay, what does the S stand for? Synthesis. And what are you synthesizing? DNA. And this is when you make the sister chromatids. And if you're doing mitosis, remember, how do we remember that? My arms, my legs, mitosis, then that's it. It's once and done. All the chromosomes are going to be lining up single file, and you're going to separate the sisters, and you have 2 cells. So you took every chromosome, copied it, and then you separate them out. Remember, if you're doing meiosis, it won't be 1 division. It'll be what? 2 divisions. And what's going to happen is first, you're going to separate the homologous pairs, and then you will separate the sisters that you made during the S stage. Okay, are we good on that? But the making of the sisters happens during S. Then in G2, we're going to be making a bunch of what? What are we going to be making in G2? Proteins, exactly. Then we're going to go ahead and go through mitosis. Now, right here, G0 is like on the freeway. It's like a what? Not a stop so much, but I'm exiting. It's an off ramp, right? And so cells could enter into G0 like through starvation and things like that. But more typically, they're going to enter into G0 because peace out, we're done dividing. And they have a set number of divisions they're going to do, and then they're going to enter G0. All right, are you feeling comfortable about that? Let's test you. Drag your dots to the correct spots. So for those of you who are not in on this pear deck right now, what you want to do is look at the G1, G2, S stage, and M, and where would you label each one of those? I'm going to try to put myself over here. Sorry, I'm going to try to move myself here. All right, so let's talk about your answers. At the end of this stage, each chromosome consists of 2 attached chromatids. What should your answer be there? S stage, right? So it should be yellow, the S stage. That should be your yellow dot. During this stage, daughter chromosomes are distributed to 2 daughter nuclei. What stage would that be? M stage, mitosis. So you made the chromosomes, and the whole point of mitosis and meiosis is just to sort out your chromosomes for your daughter cells, right? So that would have occurred then. All right, the cell doubles its organelles, accumulates the materials needed for DNA synthesis. That's got to be what? G1, so that should be red. And the cell synthesizes the proteins needed for cell division. What's that going to be? G2. Okay. Now, the next thing we talked about in the cell cycle is we talked about the control of the cell cycle, and the analogy that I gave you, I'm going to try again. Sorry, guys. The computer is trying to juggle too much. Was the air getting on an airplane, right? Because you need 2 things to get on an airplane. You need a what? Ticket, and you need to clear what? Yeah, TSA security. So your tickets are called cyclins. And I've just given you 2 cyclins here. You need to have S cyclin in order to go into the S stage. You need to have M cyclin to go into the M stage. But there are many more cyclins than that. Those are tickets that have to be present. Okay? And you have to clear security, these checkpoints. So as soon as you are, what happens during the during metaphase of mitosis? What's happening then? They're meeting in the middle. So you're stopping and you're just taking a quick second to pause and go, did all of our chromosomes line up correctly? Do we have them all lined up before we separate them? So then you would check there. G1 is the most important one. Usually G1 is making sure you did mitosis correctly, because you don't want to synthesize DNA and make more of this cell if it's been formed incorrectly. And if you can't fix the problem with the cell, what are you going to do to it? Apoptosis. And like we learned here in our next chapter, apoptosis is a normal part, right, of development. Like that's how you got your fingers. But it's also a good way to handle cells that have been incorrectly made in some way that could possibly cause cancer. Okay, that's one of 3, the cell cycle. Turn towards your bio buddy and tell him, how are you feeling about that first part? Go ahead. Is there anything you need to review there more? All right. Okay, guys, just a minute. Question? What's the difference between G1 and G2? What do you primarily do in G1? You grow and make more organelles. And what do you primarily do in G2? Synthesis of proteins. Could you do that at other times too? Yes, that's just the primary function. All you need to know is that's when you stop to evaluate that whatever stage you just did was done correctly. And you stop it, try to fix it. If you can't fix it, apoptosis. All right. Now, the next part is when we start talking about mitosis, what is the first stage of mitosis? Prophase. And what happens during prophase? Chromosomes what? Become visible. So let's talk about that. We know that the DNA, you're going to make copies of it, right? But when that is taking place, the DNA doesn't look like rod-shaped chromosomes. It looks like something you would pull out of your hairbrush, right? And so it's kind of a mess. So you want to make sure you distribute those chromosomes evenly. So it's like you're braiding them up and condensing them into these rod-shaped chromosomes. This change where you wind it on these histone proteins and on these spools, which become nucleosomes. Remember how I said it's like braids on braids on braids till you have like a braid mullet, okay, till you have that chromosome? That will happen during prophase. The chromosomes will condense so that you can see that they are made of sister chromatids. Okay, so that's that part. Now, why are you doing mitosis? We always say my arms, my legs, mitosis. Mitosis is for growth and repair. So every cell you make, the daughter cell through mitosis will be an exact clone, a replica of the original cell. Okay? And so we want to copy all the DNA and then make sure that that gets passed on. So then this for us as we grow up, like you guys are still growing, I'm done growing, but you guys probably still have growth plates and you're getting taller. Your bone marrow will remain responsive, but then pretty soon that's it for us. Plants, they have what's called apical merisms. They keep growing perpetually. And that's another story, but it's just I want to make sure we differentiate for growth and repair. Okay, next we just need to review the stages, and I'll talk you through the stages, and then we'll do the song for mitosis. You ready? So the key thing where you see a lot of action is in prophase, and in fact, this whole bit right here is all prophase. Okay, that's all prophase. Can I move myself? Can I? I made it worse. Okay, so tell your bio buddy what are all the things that occur during prophase that you remember from the song. Okay, come back to me. So we said the chromosomes, before we go much farther, how many chromosomes am I holding up right now? 2. So we said the chromosomes become visible, which means they condense. The nuclear envelope that contained those chromosomes in one location, it starts to break down. Remember, inside the nucleus, you have what's called the nucleolus. You can see it right here. The nucleolus starts to break down, so it's gone. So your chromosomes are free, ready to move around, do their little shuffle. Now, in order to organize how that happens, you have these microtubules, which is part of your cytoskeleton, that start to reach across. And they're extending everywhere out of, in an animal. Okay. Out of your animal, what are these two things? They look like churros. What are they called? Centrioles. Remember that? Now, do plants have centrioles? No. But where the centrioles sit is called the centrosome. They still have the centrosome. They still have the radiating microtubules. They just don't have those centrioles sitting in there. And where it radiates all the way around, what do we call that? Asters. Good. So now we're ready for the stage. What's our next stage after prophase? Metaphase. So in metaphase, we meet in the middle. Okay, so all the chromosomes line up single file. Then we go away at anaphase, so we separate the sister chromatids from each other. Every anaphase. I don't care if you're doing meiosis 1, meiosis 2, or mitosis. If there's some sort of metaphase, it's always there in the middle. If there's some sort of anaphase, something's going away from each other. And then telophase happens at the same time as what? Cytokinesis. And in telophase, you're rebuilding that nuclear envelope. Okay, the nucleus is kind of the opposite of prophase. The nucleolus becomes visible again. The braid's going to get undone. They decondense, so it just looks like a cell in interphase. All right, check with your bio buddy, see if they're okay on that. Is cytokinesis the same in plants and animals? No. What do they have to do in plants? Yeah, they have to build, it'll be a cell plate and then eventually a cell wall. Is mitosis the same in prokaryotic cells? Do prokaryotic cells have a nucleus? No, they have no membrane enclosed organelles. Whereas eukaryotic cells have linear chromosomes. Guys, we're up here. Prokaryotic cells have a single circular chromosome. And that single circular chromosome is going to replicate and then separate, and that's just called binary fission. And that's what happens in prokaryotes when they want to make more bacterial cells. Okay, now we need to go to the interesting here we are. Differences between diploid and haploid. Remember, our easiest definition is haploid is one set of information, which is what you would find in sperm and egg, yes? And diploid is 2 sets of information. Now, all of your body cells, what do you call your body cells? Somatic cells, right? So one set of every one of your chromosomes, one half of the set came from your mother and the other half came from your father. Are those sister chromatids, the mom-dad chromosomes, are those sister chromatids? No, those are homologous pairs. Okay, so I could have chromosome 8 from mom and chromosome 8 from dad, and what's going to happen during the S stage of interphase? Going to replicate. So I have 2 8s from mom now, and I have 2 8s from dad. Do you agree with that? And then meiosis 1, I need to separate the homologous pairs from each other. At that point, these cells technically only have one set of information. They either have mom's set or dad's set for that particular chromosome. So that's why it's considered haploid. Yes? Anaphase 1, because remember we do 2 sets of divisions. In anaphase 1, you separate the homologous pairs. And then in these 2 cells that you end up forming, in anaphase 2, that's when you separate the sister chromatids. Yes, mitosis is you copy every chromosome and they line up single file. In meiosis, you still copy every chromosome, but in the first division, they line up with their partner. Remember mom, dad, and we talked about it's like a divorce? And then you rip the sisters apart in the second division. Okay, what else? What are some other questions that come to mind? All right, so haploid, diploid. The only time you go from diploid down into haploid is when you're doing meiosis and you're getting ready for what? Sex, right? I hope that goes on YouTube. Okay, it doesn't get blocked. And you're making those gametes, those haploid cells. All right. So if we look at the overview of our life cycle, there it goes. Oh, I'm in the way of the baby, the big baby. You can't see it. Mom and dad, all of their bodies are diploid, okay? And all of their cells are doing mitosis except in their ovaries or their testes, and only there will they be doing meiosis to make their haploid gametes. Here are their haploid gametes. They only have 2 chromosomes in here, but you know all of our gametes would have how many chromosomes in them? 23, right? 23 in the egg and 23 in the sperm. You all right with that? Okay, now let me talk to you about the 23 chromosomes that are in the egg. This woman has mom, dad, chromosome number 1, mom, dad, chromosome number 2, mom, dad, chromosome 3. Yes, from her mom and dad. From this baby's grandpa over here, yes? Okay, and grandma. So when she makes her eggs, she only sends mom's chromosome 1 or dad's chromosome. She doesn't send both. She gives only one set, 1, 1, 1, 2, 1, 3, 1, 4, 1, 5, 1, 6, because she knows the sperm has a 1, 2, 3, 4, 5, 6, all the way to 23. So when the sperm and egg meet through what process? Fertilization. You form a diploid zygote, a 2N that has all 46 chromosomes. You restore the homologous pairs. Meiosis is about separating the homologous pairs first, and then you separate the sister chromatids you made during the S stage. So you only have one set of information. The sperm will provide the other set. They'll rejoin. N plus N is 2N in the diploid zygote. Okay, you processing that okay? Then this baby will grow up, right, doing mitosis to get from baby stage to adult form, and will only do meiosis in its gonads. All right, so then this slide right here is just a reminder of homologous pairs. This is a homologous pair. Even though they've replicated, it's still the homologous pair, right? It's just that each member of the pair consists of 2 sister chromatids. Okay, then when we go here, we can see the different stages. So we have interphase still, only one interphase, okay? But the S stage is still going to occur. Everybody's doubled up. Everything about prophase 1 is the same as mitosis prophase, except what will happen that increases variety. Yes, they'll do synapsis and what? Crossing over, where non-sisters will exchange part of their chromatids to kind of mix up the chromosome itself. That increases variety. It's okay because these are homologous pairs of chromosomes. They are the same size, the same shape, and they code for the same characteristics. So you're exchanging same for same. You might be exchanging blue eyes for brown eyes, but it's still what? Eyes. You might be exchanging big toes for little toe, or sorry, big feet for little feet, but it's still what? Feet. Right? So that is prophase. Metaphase, this time they don't meet single file, they meet with their dance partner. They meet in the middle at metaphase 1. And what is that called? Independent assortment. Because each homologous pair can, as long as they're in the middle, we're good. You don't need to have all the mom chromosomes on one side and all the dad chromosomes on another side. As long as you're in the middle, you're good. Okay? Then we go away at anaphase 1. So at that point, we're separating the homologous pairs. 2 nuclei at telophase 1. We've now created 2. Then we do cytokinesis. So we have 2 cells. Each one of those 2 cells, the in between is interkinesis. No DNA is replicated. It's kind of like a pause. Then each one will do meiosis 2. Meiosis 2 occurs in 2 cells. And meiosis 2 looks just like what? Mitosis, right? It looks just like mitosis because the chromosomes line up single file like they do in mitosis, right? And the sister chromatids separate just like they do in mitosis. They just have half as many chromosomes. Okay, questions? Yes. I could. Nothing. They just, same as, they're visible, but there's no crossing over because there's nobody to cross over with. And in metaphase 2, there's no independent assortment because there's nobody, you're all by yourself. It doesn't matter at that point. Okay, then we talked about variety. Okay, so variety comes from crossing over, which happens in prophase 1. It comes from independent assortment, which occurs in what? It is a meiosis 1, but what stage? Metaphase 1, good. And then you never know what sperm is going to fertilize what egg to restore the homologous pairs. So those are your 3 areas of variety. Okay, we also talked about that spermatogenesis is different from oogenesis. Spermatogenesis, that you will make 4 viable sperm when you do spermatogenesis. But when you do oogenesis, when you make eggs, you only make 1 large egg, and the others are called what? Polar bodies. And they will just degenerate. And that's because we put all the nutrients in 1 egg. We do unequal cytokinesis. And failure to separate is called nondisjunction. And that is when you don't separate the chromatids or the homologous pairs. So some have too many and some have too few. And the most common autosomal non-sex chromosome one is Down syndrome. I also have in here for you a comparison of mitosis and meiosis, how they're similar and how they're different. Do you want to go through the songs one time, or do you want to just go to the questions? Okay, so we'll do the songs fast then. I'll try to make myself bigger. Okay, and here we go. There, you ready? So we'll do mitosis, then meiosis, and we'll try to do it quickly. Ready? Here we go. And I'm gonna, you guys are far enough away. Can I take off my mask so you can see what I'm saying? Okay, here we go. We're safe. Here we go. My arms, my legs, mitosis. The first stage is what? Prophase. The chromosomes become visible. The nuclear envelope and the nucleolus disappear, radiating microtubules, asters. They meet in the middle at metaphase, away at anaphase, 2 nuclei at telophase, cytokinesis. Clone cells, yes? Okay, now we're going to do meiosis. You're getting ready for what? Sex. So you're forming haploid gametes. So the chromosomes, what's the first stage? Prophase 1. The chromosomes become visible. The nuclear envelope, the nucleolus disappear, radiating microtubules, asters. Now they meet in the middle at metaphase 1. The homologous pairs are meeting in the middle. Away at anaphase 1, separating the homologous pairs, 2 nuclei at telophase 1, cytokinesis. Now how many cells do I have? 2. And they are considered haploid because I have separated the homologous pairs. Prophase 2, right? Am I doing any crossing over here? No, because I have nobody to cross over with. Metaphase 2, am I doing any independent assortment here? No. And then anaphase 2, I'm separating the sister chromatids. And then telophase 2 and cytokinesis. And now I have 1, 2, 3, 4 cells. And I think, sorry, but can we do just the very beginning, prophase 1, first stage prophase 1, chromosomes because after synapses, did we do crossing over? Oh, I forgot to do that. So I want to make sure I had no memory of that. So in prophase 1, let's do that first part. Prophase 1, the chromosomes become visible, nuclear envelope, nucleolus disappear, radiating microtubules, asters, synapses, crossing over. Then they meet in the middle at metaphase 1 away, at anaphase 1 2 nuclei, at telophase 1 cytokinesis, prophase 2, metaphase 2, anaphase 2, telophase 2, cytokinesis, and I have 4 cells. Good? Okay, I have a bunch of questions here. If you would like some practice questions, we'll do them really fast. Okay, you ready? Highlight the stage where chromatids separate. Where do chromatids, now these are meiosis slides right here, right? Chromatids separate also in mitosis, right? So you could say anaphase of mitosis, which isn't a choice here. Anaphase of mitosis, and then which one of these? Anaphase 2. That's when you separate the sister chromatids. Straight up anaphase of mitosis or anaphase 2 of meiosis. Okay, try this one. Maybe no. Try this one, this one. So when are homologous pairs in the middle? Metaphase 1. Good. Metaphase 1. Okay, try this one. What's your answer there? Telophase 1 or straight up telophase in mitosis, right? Okay, this one. Crossing over occurs. When would that occur? Prophase 1. Yeah, remember? Chromosomes because of synapses, right? So prophase 1 is the only time when the homologous pairs snap together where that exchange is going to take place. Otherwise, they're lining up in the middle. So this should be prophase 1. Prophase 1. Does it happen at all in mitosis? No, because they never snap together in mitosis. They only do that in meiosis. Okay, and try this one. When did the homologous pair separate? Anaphase 1. Anaphase 1. Okay, try this one. For the first time, it has to be what? Prophase 1. Prophase 1, because it's the first time. Or in mitosis, it'd be just regular old prophase, yes? Well, what would this be? Telophase 2, and only telophase 2, right? Only telophase 2. Now I've switched to mitosis and the cell cycle. So what's your answer here? Metaphase, good. Is it going to switch? There it goes. Proteins are being synthesized primarily. When would that be? Pardon? When? Oh, it skipped one? I'll go back. Sorry, I try this one. Well, so when do sister chromatids separate? Anaphase. And if I was asking you about meiosis, what would you tell me? You tell me anaphase 2. Good. Okay, try. Okay, it might be seizing on your end, right? Yeah, sorry, it's a lot for my little Chromebook to handle with everything. So when are proteins being synthesized? Oh, it's going all over the place now. G2, good. When is there the division of the cytoplasm? I'll just ask them to you. When is division of the cytoplasm? Good. When does the nuclear membrane break down? When is it? Yeah. Good. All right. That was it. It just kind of seized at the end. I'm going to end it, and then I'm going to send it to you. Let's see if it will end. Okay. Do you have any other questions that you need to ask? Don't forget to do your additional objectives, right? I don't. You'd have to look on your calendar. Look on your calendar. You can always take pictures of my book. Yes.
Is this slow?
Yes.
I just have like a quick question.
Okay, hang on one second. Let me see if I can get this stop first.
Yeah.